So as Binning Singletons come in to the room, let's just see what we've got coming up. I mean, for the next hour, we have Binning Singletons. We're going to be learning a lot about them and what they do. Then we're going to go into vaccines and vaccine hesitancy, and then microbial arts and crafts workshop. So that's the next 3 hours for us on the live stream. Please, please, please feel free to engage with us. Feel free to ask questions. We're streaming live now. Wherever you're watching us, we're streaming live and you can ask us questions there. You can go to our website, or you can just ask us in social media. We'll be monitoring everything and you will get your questions answered here. And so without further ado, I would like to invite Joe James to step forward. Hi there, Joe. How are you?
I am doing well.
Fantastic, fantastic. Well, we're now pretty much in your session, in the Binning Singletons session. So if it's okay with you, I will hand the floor to you, and I'll be here kind of hiding in the background if you have any technical issues or questions. But otherwise, yeah, over to you. The floor is yours.
All right. So hi, my name is Joe James. I am the founder of Binning Singletons, which is a mentoring organization. We've been involved with a couple of microbiology associated organizations and meetings for the last few years. So today I'm really excited because everyone who's speaking today is someone who has participated in our program. In fact, I think all of them are from our World Microbe Forum program that we had back in June. So I'm going to do a very quick talk first. We actually had somebody drop out because they weren't feeling well, so I'm going to do a little bit of a quick presentation, and then we'll get to the lightning talks, so to speak. Mine hopefully will be as fast, if not faster, than some of theirs. So what I'm going to talk about first is networking and mentoring, and here we go. Hopefully. Let me pull this up. Give me 1 second. Sorry about that. Full screen. All right. Does everybody see that? All good?
Yeah, okay. Perfect.
Looks good.
So let's backwards. All right. So the title is Growing Peer Cultures, of course, a pun because I'm a dad and I like dad jokes. And so we're going to talk a little bit about the importance of near-peer mentoring. I think this is important really even for the non-micro, but maybe especially the non-micro people. Even if you're in a job anywhere really, or in school, a lot of times mentorship is really focused on that first thing on there. We have the types of mentorship, we have traditional, and there's a little bit less on coaches and sponsors. We all kind of know who these people are, but we don't always think of them in a mentoring kind of way. And then we'll talk about peer mentors, and then we'll hopefully talk a little bit about a perspective shift. So like I said, mentors can be classified in these 3 ways. You got your traditional mentors, your coaches, and your sponsors. And these are not original thoughts to me. Exactly, I took a lot of this. You'll see there's an article on ASM that I found very helpful. So that's kind of what I use some of my inspiration from. So traditional mentoring, this is often a top-down thing. You have like a senior person, a boss, a lab leader, a PI, and then you have your that person's usually an individual associated with the organization. So this is when we think of like the long-term formal guidance. So this person kind of like is your boss, your mentor, your teacher, the one who kind of gets you through everything, but you also like kind of look to them for everything in a way. And I think that's the way a lot of people look at traditional mentoring. And I think there's some there's obviously some things that can be gained from having a person that is your kind of dedicated person, but I think there's more that we're missing if that's all we do. So one thing that's nice is that they do help you recognize and achieve these defined milestones. And that success, though, is context specific. We need to remember that traditional mentors, it's really about socializing the individual to the organizational norms. A good professor in academia, quote unquote, good professor, does a really good job of churning out a lot of students. In a lot of cases, people think of that are people who would go into academia. There are obviously a lot of people who do more than that, but I think a lot of people kind of get hung up on that idea that they're preparing you for everything, and really there is a specific context that we need to look at. Moving on to coaches. Coaches are people who spend less time with these individuals, but they make a significant impact. We're all familiar, if we've all done sports, you have a coach, maybe you have practice twice a week, maybe you have a game once a week. You don't interact with those people, say, as often. Say if you're in 5th grade and you have a teacher and you see that teacher all day, that you see, your soccer coach twice a week, your soccer coach is really only focused on one thing, whereas your teacher is focused on a lot of things. So that's kind of the general idea of coaching, and I think most people can understand that. And so these coaches focus on just development of particular skills, things that don't come up as much. So if you're looking for a job, if you're writing a manuscript, designing an experiment, often learning a new lab technique is somewhere where you would find a coach. Our program, Binning Singletons, I think of as an example of coaching. So we have mentors who are really interested and excited about helping you. And there is a time frame that's involved that is not an infinite thing. It's a short duration because we help people through a meeting. And we have specific goals. We're going to help that person have success at the meeting through networking, understanding the logistics of how to navigate meetings, and then building a sense of community. So we have some definite goals, and then we have a time frame that's defined, and everybody kind of knows what that is going into it. Sponsors are your senior scientists, or if you're not in science, these are just people who have garnered substantial political capital. This is kind of the who you know part of things where mentorship is involved. So they're going to be committed to the career development of this individual and help to influence and advocate and advance mentoring. So this could be somebody who can get you on a committee. This is somebody who can invite you to a talk, to give a talk somewhere, invite you to review something for an article, invite you to a planning committee for a meeting. These are people who can help you advance your career, and them putting your name forward can really go a long way. So I want to dig into the effectiveness of the traditional mentoring for a little bit. There was a study came out in 2019, and it showed that the PI, their involvement in the mentoring laboratory activities did not significantly predict the students' skill development trajectories, which is a little bit mind-blowing if you think about how we usually think things go, which is that if you have a very involved PI, they're going to really help you with your skill development. And what they found instead was that postdocs and senior postdoc students, when they actively participate in these lab discussions, that the junior students are 4 times as likely to have positive skill development trajectories. Yeah. Oops. Okay, so these near peer mentors, they make a really, really big difference. Near peer mentees, these people who had mentors that were, so if you're a PhD student and you have a PhD candidate, or maybe a postdoc in your lab as well, those people would be your near peers, and they can really help you out with some professional and personal development. They can help you navigate things so it reduces some of your stress and then helps transition. So there's an article there that came out in 2018. So let's do a little bit of a perspective shift. And this is Beronda Montgomery has a really great article I think you should read that I'm referencing here. And this is on individual-centered mentoring, where career advancement is personally defined. So remember before we talked about normalization to organizational norms. And this is really about the personal definition of what career advancement means. So they're going to focus on the individual needs and these personal aspirations, which are not going to necessarily be the same. And one thing that she advocates, and I agree with, is developing a network of mentors. So in this case, you see that we have a lot of different types of mentor nodes and resources. So a mentor node, for example, if you look at the individual mentee and they have a nice dark blue or nice solid blue line to the one that says mentor node there, that might be their lab, the lab they're in if you're a PhD student. And some of the ones with the dash lines that you see there could be different professors that are within the same department, but maybe not necessarily your PI. Your resource node there that has a dash line there, that might be your local chapter of your scientific society. And then, there's the mentor node you see in there that's not connected. So that might be somebody that you could connect to that's in your university or your institution that you're maybe not seeing. Then the outside resources, so that's where, the mentor node on the outside, that might be, for example, you might find a sponsor out there who was your PI's PI back when they were in grad school. Or you might find something like with Binning Singletons, somebody from outside who's going to help you for a short time, and they're going to act as a mentor and as a coach instead of maybe, that traditional mentor. So in this one, I filled in a lot of these examples. I have NSERP on there, which is a really great program that helps underrepresented minorities do summer research. I have PhD Balance there as a resource node, Black and Micro, a few others. But I think this kind of helps explain and illustrate that you really need to develop this network of mentors rather than just look to this one particular person as this is my mentor and that's where everything comes from. You really need to cultivate this network of people who can help you for different things, because a mentor isn't always, somebody who's a mentor for one person in one role might act as a mentor for somebody else in a different role. So your PI, for example, might act as a coach for somebody else to learn a new technique or to help them with the job hunt or help them with writing an article. And then that's a more short-term kind of a thing. Or that person may, years from now, you may reach back to your old advisor who is now no longer your traditional mentor, but could act as a sponsor because they've gained some more capital than you and they can help you progress in your career. So that's the general idea. So my take-home advice, despite what you've seen on here, if you're a PI, don't be afraid to be a mentor. Just because there's that statistic that having the postdocs and the senior PhD students help in the lab discussions, it results in that 4-time increase in skills trajectory. That really starts with the mentor. So the traditional advisor. So if you create an atmosphere where in a culture where that kind of interaction can happen, then you're going to help foster your students' growth. And also remember, you will be a different type of mentor to different people, and that's perfectly fine. We don't need to all think of mentors in one particular way, or it's not always the same thing. And I stress this a lot, but ask your mentors what they need. Don't necessarily go in saying, okay, I'm a mentor, this is what we do, period. Here's how we help people. Really what should happen is that you need to find out what it is they want, what it is they're trying to accomplish, and then you can guide them towards accomplishing what they want and seeing what you can do to make that happen. And if you're a mentee, please tell your mentor what you need. They don't know. They, in general, are going to be acting in good faith and they want to help you. That's why they're in the job they're in, hopefully. And if you tell them what you need, most of the time I found that people are happy to help. If you are somebody who wants to go into industry, find this out hopefully before you join a lab, but make sure that your advisor can help you find somebody in industry, and don't be afraid to cultivate those mentors outside of your traditional lab structure. who can maybe help you accomplish your goals if you want to go into industry or something else. So build that network of mentors, and really your mentors, your traditional advisors should help you to maintain and expand that network that you already have. I believe that's a really important part of being a mentor. So that's all I have for now. Kept it under time, hopefully, and I will end up mine. You can email me at joe@binningsingletons.com. We're on Twitter at @bsingletons, and we have a website, of course. I would like to thank International Microorganism Day for having us, and I will move this on very quickly to Justine Debelius, who's at the Karolinska Institute. And her talk is Who's to Blame? Thinking about Disorder in Human Microbial Communities. And hopefully I can make this go as seamlessly as possible.
Awesome. I am super excited to be here. And hopefully I can figure out how to actually present my screen. It's a success already. So I love following on the heels of Joe's talk about communities, because I think microbiomes are sort of all about communities, even if it comes from a different perspective. So my niche in this is that I'm kind of a computational person, and I like big human data sets, and I like to look at them from a community perspective. So I think that there's often this view of bacteria as either good or bad. And there doesn't always seem to be space for the bacteria that sort of wants to sit at home and not get involved in superhero battles. And this good or bad dichotomy is sort of old school. It goes back to this 19th century view of how do you know an organism causes disease? Well, you have to find it in sick people. You got to be able to culture it. You have to use it to reinfect people. And then after you infect them, you got to be able to find the same thing. And it's a great approach if your tools are a plate that you can use to culture bacteria. But we're not living in the 1890s anymore. And so we need to look at this in different ways. And more recently, we've determined that a lot of bacteria are hard to culture. They don't like to grow on traditional media. They don't like a lot of air. Some bacteria need other bacterial interactions to be able to function in the community, or they don't behave that way without additional metabolites. And of course, our microbiomes are huge and diverse, and we actually find bacteria that are thought of as pathogenic in healthy communities. And that's kind of more what I want to talk about today. And so our bacterial communities exist in communities just like we do. And I want to talk about some problems that recently occurred in my local community that are similar to things that happen in your microbiome. And so we had two problems this last week. Somebody ate a very expensive pair of wool socks in my apartment of 3 humans and a dog. And then we also wanted to make a chocolate cake, and it was kind of a struggle. So when it comes to the issue of the sock, there is obviously a culprit and someone in the community who is most inclined towards sock consumption. However, this culprit would like to speak for herself. And as with other organisms in a broader community, there's a metabolic and a behavioral issue from other community members. And so she's not a bad dog. She's a very good dog. She just needs support from additional members of her community. And a classic example of this microbially is Clostridium difficile infection. So C. diff is responsible for a huge amount of hospital-acquired antibiotic-associated diarrhea, and it can be life-threatening. But the thing about C. diff is that it exists as part of many people's healthy gut microbiomes. And so it becomes an issue when the metabolic state of the community gets shifted or antibiotics get introduced and C. diff gets out of balance, just like my dog. And so in the case of a potentially pathogenic organism, it is the organism that becomes a problem, but it's also the community. A second problem at my house was this issue of chocolate cake. So we wanted to make cake. We had a recipe, and it requires 7 ingredients. And so we were going through the ingredients, and we had 3 in the cupboard. We needed 3 more. And it didn't matter who forgot their ingredient. Without one of those ingredients, we couldn't bake our cake. And so in a lot of communities, we see this loss of diversity. So it doesn't matter what organism you're missing. If you're missing enough organisms, you can't make a cake. You have problems and deficits in your metabolic state. And you see this happening in things like inflammatory bowel disease, again, sometimes associated with Clostridium difficile infection, as well as a response to certain host stimuli. And so based on these, we have multiple ways for microbial communities to get out of whack and to see weird things happen. And I'd like to conclude by saying that they're not bad microbes, just like all dogs are good dogs. They're just in a community that's out of balance. And I'll hand this back to whoever's next.
Thank you, Justine. That was really cool. I like the analogy with the dog. That was really great. My kid is in 4th grade, and she always appreciates it when people put their animals up on the calls. So that's great. All right, next is Kaylee Arnold from the University of Georgia, and we're going to talk about insects and their bugs, parasites, and bacteria. Take it away, Kaylee.
All right, hi everyone. I'm really excited to join y'all for International Microorganism Day. All right, and that looks okay?
Yep, we can see it.
Okay, awesome. All right, so yes, so my name is Kaylee Arnold. I'm a PhD student at the University of Georgia in the US. And yes, today I'll be talking about insects and their bugs, so their gut parasites and their gut bacteria. So the microbiome. So what is it? So if you're here today, I'm sure you've heard of the microbiome, or you may have heard it in health magazines, on the radio, or in the news. Well, your microbiome is just a collection of microbes like bacteria, fungi, viruses that live on and inside your body. And most of these microbes are good for you and necessary for your health. And so we have microbes all over our bodies, and inside our bodies, and I study the bacteria found in guts. So gut bacteria and other microbes are important for many processes in your body. So they can help with many things like digestion, nutrient uptake to stay healthy, stress tolerance, and then especially parasite tolerance. So the bacteria and other microbes in your guts can help you from getting sick from parasites. So now let's talk insects. So disease vectors refer to all animals that can spread diseases. Mosquitoes are the most common ones, but there are many more, including kissing bugs, which is what I study, fleas, lice, and other non-insects like ticks and freshwater snails. So these animals can spread infectious diseases like malaria, Chagas disease, the plague, Lyme disease, and guinea worm disease. And these animals are responsible for at least 17% of all infectious diseases. So now let's talk insects and their gut parasites. So many infectious diseases are spread by insects or disease vectors. And these diseases are caused by different parasites, which can be viruses, bacteria, protozoa. So now these parasites tend to live and develop in the guts of insects, and they're spread either through their saliva when they feed on an animal or human, or in their feces, as is the case for kissing bugs. And so as I mentioned, so insects, like most other animals, have bacteria and other microbes that live in their guts. Now these microbes help with many functions like digestion and even reproduction, But importantly, we study their gut microbes to understand how these microbes either facilitate or even prevent parasites from spreading to humans. So now my research is kind of focused on thinking about the parasites and the bacteria that live in the guts of insects. So my research is focused on identifying which bacteria is found in the guts of insects. And then I look at how any changes to the environment or any changes to insect behavior, so when, where, and which animals or humans they're feeding from, and how that impacts or changes the type of microbes we find in insects. So if and how any external changes impact the gut microbes of insects. And then further, I seek to understand if any of these potential changes to the type of gut microbes found in these insects may facilitate and allow these insects to spread even more parasites. So now why do we care? Well, we care because insects are responsible for a lot of infectious disease transmission, both between humans, between different animals, and especially between humans and animals. So it's really important that we study these gut microbes, we study their gut parasites, and hopefully can understand how different factors, how different environmental changes, behavioral changes impact these gut microbes and ultimately their ability to spread parasites. And then also there's a lot of ongoing research that is working to alter different types of gut microbes in these insects to directly prevent insects from spreading diseases. And then here's a photo of me. So I do work in Panama. So this is me collecting some kissing bugs out of a palm tree to eventually go and dissect and look at their gut bacteria. And that is all I have for you today. Thank you.
There I am. Thank you, Kaylee. All right. That was really cool. Thank you very much. All right, next up is Elizabeth Deyett, who is, I say from Microbigals. I believe she's actually associated with an academic institution as well, but I was on the Microbigals podcast. And here to talk about 5 tests. You coming through?
Sorry, we lost you for a second there.
Oh.
Sure. Could you just introduce the speaker?
Sorry about that. Yeah, I just got a notification that my internet connection was unstable, which is unfortunate. Yeah, next up is Tess Deyett, who I always say is from Microbigals. I know Tess is actually from, I think, the University of California, Riverside. Is that correct? Yeah, but I always say Microbigals.
That's what I say too.
Okay. She's going to talk about 5 ways that microbes affect your communication. And off to you.
Excellent. Thank you, Joe. Okay, so as Joe said, my name is Tess Deyett. I come from the University of California, Riverside, where I study plant microbiomes. But today I come to you as the founder of Microbigals and the co-host of the podcast, The Micro Moment. And I'm going to talk today about 5 ways microbes affect your communication. So keeping in with the theme. But first, I want to tell you a little bit about my blog. It's called Microbigals. We're science stories and news for the microbial enthused. And we have a whole bunch of different blog posts from history to scientist spotlights to a segment we call The Bomb, the best of microbiology, which is our favorite articles on microbiology. We also do recipes and talk about fermentation. And then recently we opened up the Society of Symbionts, which is our virtual school for microbes to learn to be good symbionts to the earth. So this is a little bit more about what we do. It's a little bit of everything from science fiction to history, to recipes to holiday favorites. So I have a lot of fun doing what I do. And with that, I just want to thank International Microorganism Day for having me, and for Binning Singletons and Joe for having me part of the session. And also to thank my team, Jonathan Mitchell, Julie Grubat, and Celia Shi, who helped me put together all of the stuff you see today. So I'm going to talk about 5 ways microbes affect your communication. And the first way is small talk, that sort of jib jabber that you have with people when you first meet them. It's usually, hi, how are you? What's the weather? What are you eating? So those are the 2 topics that people always ask me. And when I answer those 2 questions, it always has a microbial component to them. So what's on the microbe menu? We have cheese, we have bread, we have chocolate, yogurt, kefir, coffee. All of these are made possible because of microbes. And I think that is a beautiful thing. If there's a food on here that you don't like, or if there's no food on here you don't like, I would be surprised. They make some of the best food I think we have on the world. Next, I want to talk about weather. So petrichor is that beautiful earthy smell that you get after it rains. And this is actually caused by a microbe called Streptomyces. And they produce this metabolite called geosmin, which causes that smell. And then the final piece that I want to talk about with weather has to do with snowflakes. You know how every snowflake is unique? Well, that is made possible by Pseudomonas syringae, which is a microbe that may be hiding in your snowflake. So Pseudomonas syringae has a protein that's called INAZ that helps form these snowflakes into the unique structures that we see and love. So that's number 1 and 2, small talk, both in food and weather. Now we're going to move on to number 3, which is alcohol. And anyone who's ever had enough alcohol knows that it does impact your communication. Some people become angry. We don't like those people, but other people become more flirty or more loose with their lips and tell secrets. So alcohol has a way of definitely impacting our communication. And just another little plug here, I've created this microbe-inspired cocktails ebook, which is about a dozen different recipes that are all inspired by microbes. So if you'd like that, all you have to do is sign up for our mailing list and we'll send that to you. So number 4 gets a little bit more serious. We were talking about mental health. So if your mental health, if you don't have good mental health, this can definitely affect your communication. You may not want to go outside. You may not want to communicate with people. And while mental health is a multifaceted area, they are finding ways that your mental health is connected to your gut microbiome. And you have this axis they call the gut-brain axis, which is this informational highway from your gut to your brain. So as an example of how microbes may be impacting it, we have Lactobacillus rhamnosus, which can produce GABA, which is linked to decreasing stress responsiveness and anxiety and depressive-like behaviors. So my 5th and final one is halitosis, which is stinky breath. So stinky breath is another thing that can be very multifaceted. It can be caused by a number of things, but one thing that it may be caused by is a sulfur-producing bacteria that live in your tongue or in your throat. Luckily, in the world of virtual communication, this aspect doesn't actually affect your communication, but once we go back into the world again, this may be impacting your communication more than you'd like. So with that, I just want to conclude and say that microbes have a limitless potential to affect our communication and our daily lives. So just to wrap up what we discussed, we talked about how microbes are in food. We talked about how microbes can affect weather, and this has to do with small talk and communication. We talked about how alcohol impacts communication and how we have this because of microbes. We talked about how mental health can also be impacted by your gut microbiome, and microbes can affect the smell of your breath. And just one final plug, I do have another talk in an hour. So if you're free, this is Microbial Arts and Crafts. It will be here as well. And this is a collaboration, again, with about 7 other fabulous microbiologists. And we're going to do a lot of really fun things. So I hope to see you there. So thank you. And that's all from me.
Thanks a lot, Tess. I'm sorry I pronounced your last name wrong the first time.
That's okay. Everyone does. Names are hard.
I think we will definitely be back on here with my 4th grader for the arts and crafts, because I've never seen anyone love arts and crafts as much as I can. So
we're going to have some fun stuff. We have a live action drawing session. We have fermentation demonstration.
That'll be great. Okay. So next up, so everyone, we actually are going to probably finish up a little bit early. So I will try to come up with some questions for you. But if you have questions for each other, or Ben, if you want me to talk a little bit more about the Binning Singletons thing, I didn't really focus on that for my part. But if anyone wants that explained a little bit better, I can do that. But I really wanted to make sure that everybody else had their chance to talk. So next up, we have Sarah Woodward, who is at the University of British Columbia. And she's going to talk about a travel guide to the gut, how enteric pathogens navigate the gastrointestinal environment. So take it away, Sarah.
Awesome, thanks. So I'll just see if I can figure out how to share it. Okay. Can you see the right thing, or can you see my note that tells me to tell you that my name is Sarah? Does it look okay? All right, good. All right. Hi, everyone. My name is Sarah Woodward, and I'm a microbiology PhD candidate at the University of British Columbia in beautiful Vancouver, Canada. And I've really enjoyed the talk so far, and I'm going to talk to you about the incredibly dynamic and complex ecosystem we carry around with us every day, which is the gut. We've heard some great things about some of the bacterial communities that live in the gut, but I'm going to try and convince you that the gut itself, or the context in which those communities are existing, is also really, really important. So before we get started, I just want to introduce diarrheal disease to give us some context for this talk. So diarrheal disease remains a major health burden. It's the leading cause of death in children the age of 5, and every day diarrheal disease is estimated to kill more than 2,000 children. That's more than AIDS, malaria, and measles combined, and it accounts for 1 in 9 child deaths worldwide. Now, there are many microorganisms that can cause diarrheal disease, including both viruses and bacteria, and I'm going to refer to these sometimes as enteric pathogens. So enteric meaning that they're in the gut, and pathogen meaning a microorganism that causes disease or damage. So I'm particularly interested in those bacterial agents, some of which I'm sure you've heard of, such as E. coli, Salmonella, and Vibrio cholera. And these are predominantly transmitted through the ingestion of contaminated food and water. So while we know quite a bit about how these enteric pathogens cause host damage and the molecular mechanisms that they use once they reach the cells lining our gut, we still don't have the full picture. We know that ingestion of these bugs leads to disease, but we don't know a ton about the population dynamics or the way in which these bugs are actually reaching their gut niche in the first place. And to figure that out, we need to open the mystery box, which is the gut. So we don't tend to give the gut a lot of thought. It's just a tube where food goes in and stuff comes out the other end. But it's actually not a passive environment at all. It's a complex and dynamic series of parallel ecosystems. So for example, the gut physiology itself actually changes drastically from the long villi of the small intestine that are helping us with absorption of nutrients to thick mucus and deep crypts of the large intestine. There's also regional differences, from immune cell hubs to different microbial populations. And what these regional differences do is they create gradients. So down its length, we can see, for example, less undigested nutrients as you'd expect, or an increase in mucus thickness, for example, or some things that can fluctuate along its length, such as the pH or the acidity within the gut regions. In any one section of the gut, there's also these radial or cross-sectional gradients. So if you imagine looking down the middle of a pipe, for example, there's an increase of oxygen closer to the cells that line your gut compared to in the middle. So it's unsurprising then that enteric pathogens have evolved to use the presence or absence of these regional signals to determine where they are in the gut, or even what stage of disease the host is in. And this tells them, importantly, where they need to go and what they need to do when they get there. So some of the behaviors that are directed by sensing these gut signals include growth of a population, motility or moving to a particular area, attachment to your host cells, and even virulence, or those host damaging behaviors such as hijacking our host cells. But the good news is that because of all of these regional differences that bacteria have to navigate to cause disease, the gut is actually highly restrictive. For example, major barriers to causing disease for an enteric pathogen include passing through the highly acidic stomach acid, which is able to kill a very large number of bugs. There's also peristalsis, or that luminal flow that helps us not only push our food along the GI tract, but also helps to flush away bacterial pathogens. And of course, those resident microbes you've been hearing about, which are able to resist colonization by enteric pathogens, in some ways just by taking up real estate and nutrients in the gut. So you can think of these each as barriers or literal hurdles that pathogens need to overcome in order to host and cause disease. If we can identify and manipulate these barriers or even alter the regional signals that pathogens respond to, this can allow us to better prevent and control diarrheal disease. But understanding gut barriers to colonization can even help us to improve probiotic therapies, or that is the introduction or colonization of those so-called good bacteria, which we have a notoriously difficult time getting to stick around in our gut after we've ingested them. So I hope I've convinced you to think a little bit more about your gut and why it's important to understand not only how the enteric pathogens damage our host cells, but how they get to those host cells in the first place and whether we can intervene in the process. So with that, thanks for listening. Special thanks to Joe and Binning Singletons for the opportunity, as well as the International Microorganism Day team. And of course, I'd also like to thank my colleagues in the Finley Lab, where I'm doing my PhD here in Vancouver, including Jorge Peña, who's going to speak next. So, and if you have any questions, feel free to reach out to me. Awesome.
Okay. Yeah, with that, we're going to go right to Jorge, which actually is here on my screen, but I don't know where he is on your screen. From the University of British Columbia, and title is Everybody Talks, Bacterial Communication in the Gastrointestinal Tract, which I don't know if Everybody Talks is a reference to a song title, but if it is, I get it. So
I'm impressed you got the reference. That's pretty. Thank you so much. Am I sharing already? Can you see everything? Okay. Okay, just give me 2 seconds. Okay, well, thank you so much, everyone. And like, as just like said, I'm going to talk to you a little bit more about like bacterial communication in the gastrointestinal tract. Jorge Peña, and like, I'm happy to talk to you. So like, thanks everyone from the International Microorganism Day and Binning Singletons for like inviting me to give this little talk. So I don't know why I even had to give like an introduction at this point, like with all the beautiful talks that we just heard. Like, I think that's not much that I can say, but as we have heard so far right now is that like everyone, we know that bugs can just be found everywhere. And like, since microbes can be so well adapted, they basically can be found on any environment that you can imagine, from like leaves from a plant, their roots, the soil, to your like skin, any environment in earth that we can hide, even our food, as like we have heard from Tess, like our cheese, our coffee, our beer, to like even the most unhospitable like environments, such as like the bottom of the ocean floor, the permafrost, even in volcanic environments too. But one thing we don't talk too much, or we don't hear too much, as Justine said, is that like bacteria are just not living on isolation. They actually live on very complex communities where they can actually be interacting with each other, where they can even be like cooperating, so they can actually help each other to then actually thrive in this like really like harsh environments. Or they can even be competing, so they can actually, like as Sarah was pointing out, thrive when there's very hard competition to like actually colonize a niche. They need to compete so they can actually thrive. Or even cheat, which I found hilarious. They can actually like take advantage of others so they can actually flourish and establish. Now, since they live on really complex communities, it's been found that they can actually talk to each other. At least some bacteria, especially like gram-negatives, can do a process known as quorum sensing, which is a way of bacteria-bacteria communication. And when they grow, they can secrete really, really small molecules. And as they start growing and accumulate more in the environment, they can use this as little cues that allows them to respond what's happening around them. So it could be a cue of like, who's around me, or like, are there many of my brothers and sisters around me, or is there a lot of people there I need to compete with? And then use this as a cue to then regulate different group behaviors, such as like production of like toxins, or like motility, like the production of biofilm formation, bioluminescence, and others, which would be like behaviors that are very expensive for the bacteria to produce by itself. But when they group like work together as a team, then it's something that it can be like a competitive advantage. But now, as I told you like earlier in my first slide, like we know that bacteria lived in like many complex communities. And we know that like they can talk about, like they can use quorum sensing or like this communication signals to talk with each other. But are they actually talking in our gut? And it's something that is like still not very well like explored. And it's very interesting because like the human microbiota is still one of the most richest and most densely populated communities inside of our body, but still we don't know actually like who are they actually like these molecules or like produced, or can they be secreted, where are secreted, what they're actually doing. And there's many questions that I'm trying to like answer in my PhD, but we do know that actually they can be measured and detected in the guts in very like and different amounts. Then as well that it's believed that they can play a role to like actually contribute to our health and maintain like a healthy composition of our gut microbiome. So now, why it's so important to like explore this like communication signals in our gut? And it's because they can actually confer like a really interesting approach. For example, some pathogens can use these little communication signals to like coordinate how they can cause virulence or harm into ourselves. So if we can understand better how they can use these signals to like modulate this virulence, we can like maybe make different like treatments that can cause like the inhibition of this communication signals, not allowing this bacteria to mount these group behaviors and just act them as individuals, and then just allow like the immune system to naturally clear this infection instead of having to use antibiotics. Or at the same time, we can use them as a different way to like to have a disease microbiome that it could be like associated with disease, and then help them to like push the creation of a more healthy or a more balanced microbiome by allowing different like microorganisms to thrive and like grow. So with that, I hope that I can like at least convince you, and like takeaways from my talk is that like bacteria can live in very complex environments, and they can actually be talking to each other and responding to what's around them. They can use this small molecules known as autoinducers to like communicate with each other. And as well that like exploring and like these tools can offer a tool to then use it to like use this as a tool to like I don't know, like I'm lost of words, but to like incorporate it as like another molecular toolbox to like create healthy environments. And with that, thank you so much for everyone, especially everyone in the Finley Lab who has let me explore a lot of the questions that I like proposed today, and as well to Binning Singletons, like showing a very nice environment for me to show my science, and as well my International Microbiome Day for like a place to like express my talk. So thanks so much, and if you have any questions, please feel free to contact me. Thanks so much.
All right, thank you very much. We do have a question for Tess about the microbe-inspired cocktails, and what is the most alcoholic one? Tess, do you know that answer?
I do, yeah. So we made a cocktail, and it's called Toughest Tardigrade, and it is a beer float cocktail that includes a shot of vanilla, or 2 shots actually, of vanilla vodka inside a 15% stout. So it's fairly alcoholic, and when you mix your alcohols, you will definitely feel it a little bit faster, which is why we named that one Toughest Tardigrade.
That's pretty great. So this was suggested. I think it's actually hopefully a good one. Well, let me give a little bit of a background first. So Binning Singletons is something that we came up with back in 2019. Actually, I came up with it a year earlier and wanted to try it, and then we actually did it in 2019 at the ASM Microbe meeting. And so the idea was that a lot of people go to these. I don't know if you're not in science, you probably have been to some kind of a meeting or conference with people from your industry, and you get there, and there's just all these people, and you don't necessarily know them all, but you may have heard of some of them depending on your field, and you might be intimidated because honestly, when you suddenly show up and there's thousands of people, it can be a little intimidating, and imposter syndrome can really kick in. So our idea was to help people kind of jumpstart and raise the floor of what their meeting experience is. So rather than having to go through those first 2 or 3 times on your own and learn a bunch of stuff kind of the hard way, and maybe have negative experiences and never come back. We want to make that first experience pretty good. So the idea was, so we used a lot of puns from microbiology, and so I know that this audience is really the microbe interested public, not necessarily microbiologists. So our puns kind of go hard in on the micro, so they're not necessarily as useful as an explanation. But essentially what we do is we want to group together. So if you've gone to one of these things and you've ever felt left out, or even, I think we've all felt this if you've been a new kid at school, or you got to high school and you had different friends and whatever. What we try to do is give people a community, so group people in groups of like 2 or 3, and then give them somebody who has done the meeting before as a mentor to kind of help guide them through it. And so that would be our Binning Singletons idea. The next thing we do is what in our description is called horizontal transfer, which comes from the fact that microorganisms often can transfer genes from one to another that did not evolve within that line. And so this is where sometimes you get bugs that acquire an antibiotic resistance that they didn't have because they're around something else that has that. So suddenly an organism that wasn't really that bad before, now you get an ear infection and you take your antibiotics and it doesn't work because it has got this horizontal transfer of this nice gene. But for it, it's a nice gene. For us, it's not. So our idea is that this horizontal transfer takes place between both the members, so the peer-to-peer kind of mentoring kind of thing, but also that the more experienced person can offer their advice. Also importantly, unfortunately, we didn't get to do this at the World Microbe Forum, but we kind of did it virtually with some of our Zoom calls, is you want to bring all these people together who feel this way, and it helps people realize that they are not the only ones who feel this way. And it's like actually some really, really cool, smart people that maybe you were intimidated by are also there. And you're like, oh, well, okay, cool. I mean, I'm in this group too, so we're all good. And then the next we jokingly call it the exponential growth phase, which if you are growing bacteria, there's a phase where they just grow and double every, if you're E. coli, you can double every 15 minutes under the ideal conditions. So, you suddenly have a whole bunch more E. coli than what you started with. So our idea is that these people now who've worked together and kind of spent a little bit of time together now can really grow at a much higher rate than they would have before. And so really that's kind of the general idea of what Binning Singleton is. Again, for me, it was really as a result of having gone to many, many meetings by myself and feeling like I didn't really belong there and everyone else was smarter than me and they knew what they were talking about and I was faking it. And so it was kind of a way to help get over imposter syndrome. I don't know if you can see right above my head. There's a quote by Charles Darwin back there that says, but a lot of you have heard it. It says, but I am very poorly today and hate everyone and everything. One lives only to make blunders. And I have this up in my office because if Charles Darwin can have that day, then I can have that day. And I have that day a lot. So I think it's important for everyone really to understand that, even if you feel like sometimes you're not measuring up to what some idea of what you think a scientist is, that actually, or if you're in another field, whatever your profession is, you're probably doing better than you think. And if you find a community of people that you can share with and talk with, you'll find out that you're actually probably doing a lot better than you think, and that it's really a lot more fun if you go through this together. So I very cordially like to say that we are cultivating a culture of community. Again, cultivating culture, all those nice bacteria jokes. But so one question somebody wanted to ask is what kind of impact Binning Singleton has had on your route. I'm not sure I like the question too much because I don't know that I want to take a lot of credit for really changing anyone's life, but hopefully people have had some small benefit from being with us. So if anyone wants to jump in on that, and then I'll also take other questions and pass them on. Anyone? Justine? Oh no,
I was just saying it's awesome. Two thumbs up.
Oh, Kaylee has her hand up. Okay. Kaylee.
Sorry.
I'm missing Zoom responses. It's my fault.
Well, so I'll just say quickly is that, so I'm kind of new to microbiology. I was trained as an ecologist and in marine bio. So kind of just having this avenue to have like less imposter syndrome and kind of feel a little more kind of connected to like microbiologists has helped a lot.
I want to like say too that like I've been to like a conference before, and it's kind of like so challenging when you don't know anyone and you're literally like the one odd one in the room, and you just, I mean, I didn't even know how conferences work. So like it makes such a difference when like you can have like a community to thrive, right? Like as I like said something on my talk, like that makes a huge difference. And it was so much easier to like, oh, I want to talk to this person or whatnot, like having like that group as a support. And the other thing is it just opens like opportunities too. Like I feel like it helped me to expand my social like media and like reach, and like as well gives you more opportunities, such as like that's why I'm here, right? Like, so thanks.
Yeah, I'll just echo that. I think, I mean, when I heard about Binning Singletons, I absolutely fell in love with the idea. And I think sometimes when you hear these ideas, you can think, well, you know, maybe they're not actually going to give you what they're saying and it's all just talk. But I going to the World Micro Forum and being part of Binning Singletons, they had actual moments and networking hours where you could talk to people. And it was just so nice to hear other people say like, you know, I'm super introverted, I hate talking to people, and just being able to relate to other scientists who also feel the same way. It just makes you feel a little less alone in the virtual world.
Yeah, I have nothing original to add to that, but I agree with everything everyone has said. It's been a fantastic networking opportunity, and even just kind of going to those other broader conference networking events as a little Binning Singletons team was key, because otherwise I don't think I would have gone to them. And just like learning how to navigate some of the huge conferences like that, it's been fantastic. So yeah.
I particularly enjoyed when we all kind of descended on Johnny Chang as he was drawing art. So I have my shirt that he drew for me. You saw in Jorge's presentation, he also had one. I think like at least 50% of the people in that session are from Binning Singletons, because I made sure to share it. So that was pretty great.
That was kind of crazy too, because I had to wake up like at 4 a.m. to like go to the
Yeah, I actually thankfully had gotten mine at the session the day before, because I had something else I wanted to do and I couldn't get in, because even though I got up at 6, I still couldn't get in because he's such in demand. But his art is awesome, so it's worth it. Okay, so Ben, do you have anything that you want us to talk about, or do you have any questions that we haven't seen? Because I'm not seeing YouTube or any of the other formats.
No, we don't have any questions coming in at the moment, but we've got how long is it? A couple of minutes left. So I guess are there any more questions from you guys? Is there any kind of final remarks? Otherwise, we can close the session. It's been absolutely fantastic, I can say. I've really enjoyed listening to you all and kind of learning more about Binning Singletons. It's really inspiring. So thank you for that. Are there any kind of closing remarks? Anything anyone would like to say?
I know that I can say that having these kinds of presentations where, you know, junior early career people are presenting their work, as somebody who's been in my job for 21 years now, it almost increases my imposter syndrome, because I'm like, oh my God, they're doing things that I can't even imagine, and it's amazing and really awesome and cool. So I think it's really good for, because I know a lot of us, now if you haven't been to a lot of conferences, you maybe haven't felt this, but sometimes if you go to the similar conferences, you kind of hear the same talks from the same people over and over again. And I think it's so hugely important to get those talks in where the junior people, the early career people, get to talk about something that they're the ones working on it in the whole world, because it's a small little thing, but it's like their central focus, and they're going to teach you something you don't know. And I really hope that in the future I know that ASM has done a pretty good job of this, of adding some talks for some junior people, but I really hope that a lot of conferences go this way. I know that you've got the headliners and people want to hear the big hitters, but there's some really, really good work being done out there by the really career people, and I would love to see it. So that's my little soapbox.
I can also say something maybe. So I was the one asking the question on what was the impact of Binning Singletons on your route. I didn't know it before. So I have to say that I find it amazing. I want to join, but I'm a postdoc. I feel I'm a bit too old to be a mentee, too young
I'm a postdoc.
to be
I was a mentee.
Yeah?
Yeah.
Okay. Maybe it's not so...
We had a tenured professor as a mentee one time. So
really? Okay.
Yeah. No, I mean,
So maybe I think I'm more of a mentee than a mentor, but I have to say I totally relate to the feeling of you're the odd one, or you don't know how to act, or if you can approach someone, or you feel insecure about saying something. Everyone feels like so much smarter than you. So great initiative. I just wanted to say this. And I also want a t-shirt. They are cool.
So this one is a one-off. I have the only one of these, but
that's the original one, I feel.
Well, this one's different than all the other ones. I think you can see Sarah and Jorge, the ones we did for World Microform. I actually have one that I have whipped up for International Microorganism Day, but I have to clear the logos with IMD folks to make sure I can put it on there. So, but actually, I can show it real quick if anyone wants to see it, because I think it's
let's see it. Let's see it.
Sure.
Where did I put it? All right, so there it is.
Oh, nice.
So I've got the Binning Singletons microbes on there, and then I put the International Microorganism Day microbes on there as well.
That's nice.
So I actually have something just, I broke something. It went away.
No, it's fine.
Very cool.
So yeah, I will totally look up to it.
So that's what. Oh, and then I did put my oh, where'd it go? Yeah, I put my it's a front and back shirt, and so I have my, where's my share screen? There we go. Okay. There we go. Yeah, on the back I put my nice cheesy slogan of cultivating a culture community. But again, you can see I have the logos there, so I got to make sure that that's actually kosher before I actually print it.
Excellent, excellent. Send it over. I'm sure it won't be a problem, but yeah, it'd be very nice to see that on a real t-shirt. And yeah, with that, I'll say thank you again to everyone. Thank you very much, Joe, for sharing, and thank you everyone for your amazing talks. And I guess we'll move on now. It's that time of the evening. I was flown by. It's absolutely flown by. And now we're going to move on to the next session, which is vaccines and vaccine hesitancy. And